So I'm going to be talking about the function of sleep from the perspective of how different animals sleep. And then I will be talking about the peptide with two names, hypocretin and orexin, that was discovered in 1999 and has become the focus of research on narcolepsy. So studies of sleep function have commonly relied on sleep deprivation. I won't really be using that technique, and the reason is that this is a problematic technique. Interference from the subject's learning during the deprivation period degrades recall of items learned prior to the start of the deprivation. So this degradation can be attributed to sleep loss, but it can also be attributed to a standard, well-known learning phenomenon where learning interferes with prior learning. Also, during sleep deprivation, cortisol is released, and cortisol release alone, even in waking, diminishes recall of items learned prior to the cortisol release. And unfortunately, sleep deprivation studies, which are numerous, have generally not controlled for these two factors. An alternative to sleep deprivation studies of sleep duration in a variety of species can provide insights into sleep function because non-REM and REM sleep amounts vary over more than an order of magnitude between species. These differences can be correlated with behavioral, cognitive, neurological, and dietary, as I will be emphasizing, differences without introducing any stress. So this is a graph of sleep amounts in all mammalian species that have been studied, broken into three groups: carnivores, omnivores, and herbivores. So the y-axis here, the up-down, is number of hours of sleep, and the x-axis, left-right, is the size of the animal, the weight of the animal. And so I think many people have the impression that humans sleep more because we're smarter. But in fact, if you look at the carnivores that have been studied, the little red box shows the human sleep time, which is the lowest of any carnivore that's been studied. Of course, humans can be considered any of those three categories. Among the omnivores, human sleep is also quite low, but there's a cat that has slightly less sleep, a type of cat. But finally, herbivores, you see a very nice regression of sleep and body mass, and humans fit in perfectly in the middle. Basically, bigger animals have less sleep. And you'll see this when I discuss our studies in the elephant. And so humans fit in. We're sort of a medium-sized animal. We have a medium amount of sleep. Now, I'll be talking later about sleep in hunter-gatherers. Human hunter-gatherers who we've worked with spend most of their day looking for and consuming food. They're not sitting around. They spend their time doing what humans have always done, spending a majority of the daytime looking for food, as is the case with these other animals. But we're kind of oblivious to this because the development of agriculture has basically eliminated most of what humans used to do during their waking period. Because instead of spending 12 hours finding food, we go to a supermarket for a half hour and that's it for the week. So there had been studies of sleep in elephants in zoos, but never in the wild. And so I had an opportunity to study sleep in African elephants in Botswana with my buddy Paul Manger. And so here you see a plot of sleep for 35 days in 2 elephants. And you can see, first of all, I can't quite read the dates here, but there was a date early in May where matriarch 1 didn't sleep at all, and early in April rather, and then there are 2 in May where this elephant didn't sleep at all. And likewise in matriarch 2, an interesting thing is that there was virtually no rebound. There was no increased sleep to make up for a day without sleep. So African elephants in zoos sleep 4 to 5 hours a day. But when we summed up all the data we have, African elephants in the wild sleep only 2.1 hours a day, the lowest of any land mammal. Despite the difference in sleep, or maybe because of it, wild elephants are healthier and live much longer than elephants in captivity. Elephants have impressive cognitive abilities. The saying is, the elephant never forgets. And their lifespan is between 60 and 80 years, similar to human lifespan. And a key item here is each elephant eats about 800 pounds of vegetation a day and walks 10 miles a day. And these two things are obviously related. Elephants travel in herds of 10 to 15 elephants. So if you talk about the herd, they're eating 8,000 pounds of plants a day. And in order to do that, they obviously can't stay in one place. So they're migrating continuously. And they also have to remember where they were last time because they don't want to go back to a place that still hasn't regrown from their eating there. One of our elephants walked 41 miles in one day. It's kind of amazing. A marathon is 26 miles. So days without sleep are not unusual, not followed by rebound. And most sleep, that's the bottom graph in C, occurs toward the end of the night. So at the other extreme, we have the little brown bat, which has the most sleep of any animal, sleeps 20 hours a day. Now there's no reason to think they have any particular cognitive powers that other animals don't have, but they are specialized for eating moths and mosquitoes. And moths and mosquitoes generally come out at dusk and then they go back to their nests. So there would be no point for the little brown bat to be flying around at other times because they would be expending an enormous amount of energy. So it would be maladaptive. So they have evolved to spend most of their day hanging at the side of a tree or in a cave and just coming out when the mosquitoes are out and then going back to sleep. Now, this is a figure I first made in 2001, and I updated it for a review in 2022. So, confronted by this kind of data, people say, well, really, it's REM sleep that's important for cognition. And so I made this chart. All of the animals on the left side of the chart have more REM sleep than humans, while the animals on the right side have less REM sleep than humans. Also, you can see that all the animals on the left side have more total sleep, not just REM sleep. So sometimes REM and non-REM are considered unrelated and completely functionally different, but in fact, REM is correlated with non-REM. The amount of REM across species is very highly correlated with the amount of non-REM. So there's a linkage, and I'll get back to why that might be. The animal with the most REM sleep is the platypus, so-called duck-billed platypus. You see the ferret and the opossum, the big brown bat, also a little brown bat would fit in there. And on the other side, animals that sleep less that have less REM sleep than humans. You see the guinea pig and the guinea baboon. Apart from both living in Guinea, there's not much in common that you would think would cause them to have the identical amounts of REM and non-REM sleep. The guinea pig is a rodent with a small brain. The guinea baboon is a primate with a large brain, a lot of cognitive abilities. And then on the lower right is the bottlenose dolphin. The bottlenose dolphin doesn't have any REM sleep. In fact, all cetaceans do not have REM sleep. Cetaceans being whales and dolphins. So many of you may know dolphins have this unusual form of unihemispheric sleep. But the unihemispheric sleep is non-REM sleep. It's sleep with slow waves in one hemisphere and then sleep with slow waves in the other hemisphere. There's never anything that resembles REM sleep. So this is platypus. Platypus is, does not live underwater, but he hunts underwater. They're very cute, but they're actually quite dangerous to handle. So it's good to have a graduate student around. Who in my case was Paul Manger. He also did the elephant study with me, so there's a thread running through that. The bill and the platypus is electroreceptive, so they go side to side scanning the environment, and they're able to pick out prey in muddy water because they don't need to see it. They can get the electrical impulses from the muscles and hunt in that way. So this is what we see with a conventional polygraph recording. You see it in the bottom two sections. You see bursts of rapid eye movements, some accompanied by twitches. You can see the EMG activity on the bottom three traces. There's two rapid eye movement periods with twitches and one without. But basically, there is a mixture of REM and non-REM, and I think this is the primal form of sleep. I think what happened is, so these are egg-laying mammals. They haven't changed for nearly 100 million years. I think it used to be that you would alternate activation and inactivation in the cortex, you meaning animals. And eventually what has happened as this is differentiated into periods of activated EEG and periods of slow wave EEG. But that's just a logical supposition. There's no way to go back and study the physiology. But that's what they have, this mixture, both the echidna and the platypus. Those are the two egg-laying mammals found only in Australia or adjacent areas. So the platypus and the echidna have more REM sleep than any other animal. And I will show you, hopefully. Oh, there we go. Okay, great. So you see the animal is moving its head side to side. In the pool, this is done in Brisbane, in the pool are these animals called yabbies. They're crustaceans that the platypus likes to eat. And it crushes them. And here's REM sleep in the platypus in the burrow. Okay, so I wondered, as you might, why do monotremes have so much REM sleep, 8 hours a day of REM sleep? Their brains are fairly small. but their body and brain temperature is only 31 degrees centigrade, 88 degrees Fahrenheit, in contrast to our 99 degrees. And I wondered if brain temperature might be inversely correlated with REM sleep amount. That's not usually the way it's been thought of. But part of the reason I thought of that was work that Al Rechtschaffen did in 1989, where he did this series of studies of sleep-depriving rats. And what he found is if you sleep-deprived rats, they get cold. And then when the sleep deprivation starts, there's an enormous rebound of REM sleep, regardless of whether they've been deprived of non-REM or REM. So there seemed to be a temperature clue there. It was just a stab at a reason for why the platypus and the echidna have so much REM sleep. But then I was asked to review this work a couple of years ago, and I thought, well, maybe I can push this temperature thing and look at a whole bunch of animals and look at their temperatures. So homeotherms, animals that regulate body temperature, can be categorized as birds, placental mammals, that's us, marsupials, and monotremes being just the echidna and the platypus. And you see the core temperature in red here of these two groups of animals differs, with the highest core temperature in birds, then placentals, then marsupials, then monotremes. Also, I continue to be amazed that within each group, the temperatures are quite similar. So among placentals, for example, 37 degrees or 98.6 is our temperature. That's also the temperature of the mouse, the rat, the cat, the dog, the elephant. It's all the same temperature within that group. Similarly, birds, the ostrich and the emu and the parakeet all have a body temperature of 41 degrees. And the monotremes are 31 degrees. So that is quite amazing. And then I looked at all the data that had been gathered on species in each group and calculated what the average amount of REM sleep was. And what you see is the group with the highest body temperature in all its members has the lowest amount of REM sleep. All birds have REM sleep, but very little. And placentals have more REM sleep, and then marsupials and monotremes. And if you correlate across groups in this way, the correlation between body temperature and amount of REM sleep is negative, but it's an almost perfect correlation, minus 0.975. In other words, if you know what group you're talking about, you know the average of the REM sleep, almost exactly. Now, if you look in the brain, you see further evidence for this temperature relation. So here you see at the top channel, we're measuring the hypothalamic temperature, then the pontine temperature, and then EMG and EEG. And what you see is temperature falls in non-REM sleep. This is because the brain is less active. It's using less energy, which is highly adaptive. The reason you go to sleep is to save energy. But when the temperature falls significantly, REM sleep kicks in and raises the temperature. It's very much like your home heater at night. You know, you go to sleep and the room temperature goes down, and then the heater goes on, brings the room temperature up to, 70 degrees or wherever you set it, and then stops and it falls again. And that's the cycle. And this is what REM sleep does for the brain. Now, Michel Jouvet, a famous French physiologist, did a study. I don't know if it was really a study, but he published this figure, which may have been an accident, but he was studying the effect of disconnecting the brainstem. We know REM sleep is generated in the brainstem alone. Actually, that was Jouvet's greatest achievement. He showed that you don't need the forebrain. Literally, it can be removed from the body, and you get REM sleep generated by the brainstem. But what doesn't happen is there's no thermoregulation, because if you disconnect the hypothalamus, where the temperature sensors are, from the brainstem, where the motor control is that can adjust temperature, the animal's temperature will just fall. Usually you would have an animal on a heating pad, and I don't know if in this case the heating pad didn't work, but what happened is the cat, which generally has 15 to 20% of the time in REM sleep, the cat's body temperature fell all the way to room temperature, and its REM sleep amount increased to 80% of the time in this very linear way with the fall of temperature. So again, a spectacular example of how the temperature is controlling REM sleep. So cold monotremes, the platypus and echidna, and cold brainstems generate lots of REM sleep. Another idea that I see on the program will be discussed later in the meeting, but is this idea that Makeen Nedegard at the University of Rochester proposed, that the function of sleep is to clear brain toxins, and that brain toxins are cleared at a fast, higher rate in sleep than in waking. And so this generated a little controversy, and the most recent word on this was by Nicholas Franks, who is in London, and his conclusion was exactly the reverse. So I think the idea that sleep is removing poisons from the body is questionable, but these two papers, which you can read for yourself and make up your mind, but I think Franks is closer to the truth. I did a series of studies in hunter-gatherers, because there's this perennial argument in the sleep field about what natural sleep is, particularly the idea that we're all sleep deprived because we have electric lights, not to mention TV and cell phones, and that really we're meant to go to sleep at night at sundown and wake up at sunrise. And because we've generated this artificial environment, we're sleep depriving ourselves. So I worked with a bunch of anthropologists on this study. We looked at three groups. One was the Chimani in Bolivia, and then the San group in Namibia, and the Hadza group in Tanzania. And I can give you one conclusion up front, is they all have the same sleep pattern. So very much supports the idea that there's sort of a common pattern of sleep. It's not idiosyncratic for one location or another. So this is one of the data analyses we did. We had a weather station so we could track the temperature, which you see in the dark blue tracing. That's the environmental temperature. These subjects all swallowed temperature pills, which transmit core temperature to an outside receiver without otherwise disturbing them. And so that's the red line is the body temperature. You can see, interestingly, just looking at the environmental temperatures is something we all know already. But after sunset, temperature goes down, and it goes down in this sort of regular way. And then at dawn, which is when these individuals universally awaken, the environmental temperature starts going up. And this is unlike the way most of us sleep. Most of us sleep at a constant temperature, which is maybe lowered, but it doesn't track the natural fall of temperature. And even if you set your thermostat for a lower temperature, which is good advice, it's still not simulating what happens in the wild, what people sort of evolve to live in, this environment of gradually falling temperature and gradually increasing temperature. So the first point to make is you see at the top in the middle, there's sunset and onset. So that's sunset and then onset of sleep. And then the black bar is the sleep period. So they do not go to sleep at sunset. No hunter-gatherers go to sleep at sunset. And the sleep duration is 7 hours in this sample, and actually in most of the other samples we took. There's this historian, Roger E. Kirch, who has gotten a lot of publicity. He wrote a book essentially saying that you're meant to go to sleep and then get up in the middle of the night for a couple hours and then go back to sleep. And he based this on, he's a historian, he based this on the literature of sort of medieval Europe, that the phrase first sleep and second sleep were used. Now, I don't doubt that some people go to sleep, wake up, and then go back to sleep. I mean, that's kind of a sleep pathology, common sleep pathology. But we didn't see that at all. We saw no individual who was having that kind of sleep. And also, we looked into insomnia. We asked the individuals in their native language if they ever have trouble sleeping. And we also had our active watch records of their continuous sleep. And they almost universally said, "What are you talking about, having trouble sleeping?" Actually, the guy I was working with named Oma, who is a San tribesman, said, "Oh yeah, Jerry, 3 years ago I broke my arm and I really couldn't sleep because it hurt so much." So I said, "Well, that's not exactly what I was getting at, but thanks for the information." So this is now a chart of sleep for an entire year in these San hunter-gatherers. And you see the average is about 7 hours. There's more sleep in the winter than there is in the summer, about a 50-minute difference. Of course, the months are reversed. This is all south of the equator. So we did a total of 8,000 days of recording in these 3 hunter-gatherer groups. They all had similar sleep parameters, no sleep onset at sunset, even in the absence of electric light. Average sleep onset was 3 hours after sunset, which is not that different from what we do. And their behavior is not that much different from what we do. We sat around talking, singing, dancing, planning for the next day, and eating, of course. Sleep duration in these societies averages 7 hours. If anything, less than we do. Other diurnal primates like chimps sleep more than we do. There's little napping. We included that in the totals we're reporting, the 7 hours. But there's very little napping in the winter. But in the summer, about every 5th day, there would be a nap on average. And I already mentioned 50-minute difference between summer and winter. So this is not my work, but this is this paper by Kripke that he published in 2002. And this was based on an American Cancer Society where they studied, they gave a survey to 636,000 women and 480,000 men. They were trying to get at behaviors that might predispose to cancer. And so they surveyed foods and exercise and sleep time. And because this is such a huge database, Dan Kripke was able to take advantage of that and ask questions about sort of what is the healthiest amount of sleep. What was done is a survey was given, and then 5 to 7 years later, it was identified who in this group of over 1 million people had died. And that was essentially what's plotted as a function of sleep time. And what you can see in the gray boxes in the bottom two frames here, both in men and women, that the healthiest, the sleep amount that predicted the longest lifespan was 7 hours, exactly what we've seen in the hunter-gatherers. And that people who slept more than that had a shorter lifespan. And I think a lot of people in general population now think the longer you sleep, the longer you're going to live, the healthier you are. But of course, if you're sleeping, one trite explanation is you're not exercising, and being inactive has its own downside. But whatever the explanation, the agreement is 7 hours of sleep is best for lifespan, and the shortening of lifespan is relatively rapid if you sleep more than that, but not so rapid if you sleep less than that. It doesn't really get to critical levels until you're sleeping, or you think you're sleeping 3 hours, which is very rare. As you can see, the standard error reflects the very few people who actually have, who report that amount of sleep. Okay, now I'll talk about something more clinically relevant. So in 1999, two groups of investigators, geneticists, discovered what turned out to be the same peptide. One group, led by Sutcliffe, called it hypocretin, and another group led by Yanagisawa called it Orexin. That's why two names remain for this one peptide, but they were talking about exactly the same thing. In 1999, Cimelli worked in Yanagisawa's group, was studying knockout mice, that is, mice whose genetics were manipulated to prevent the synthesis of this peptide. And what he saw this strange behavior where they seemed to cease movement periodically, and he thought that this resembled cataplexy. And this was a very exciting finding because nobody knew anything about the cause of narcolepsy. Some people even would say it was a psychosomatic disease. So our group and Minot's group immediately went to work, and within a year we had both found that there was a loss of hypocretin cells in human narcolepsy. And so it seemed that the mystery of narcolepsy had been solved. I'll muddy the waters a little bit as I go through these slides. But this is our data. You see in the center is a schematic drawing indicating all the cells in a normal brain and all the cells in a patient with narcolepsy, all the hypocretin cells, which is the word I will use to describe this peptide. And you see the immunohistochemical data that this is based on in the left column and the right column. So we then developed techniques to record the neuronal activity of hypocretin neurons that involved being able to identify hypocretin neurons among the other neurons in the hypothalamus and allow them to freely behave, and we could see what behaviors they were related to. So I will show you the video, but since it goes by fast, I want to direct you to what to pay attention to. There's 3 channels displayed like a regular polygraph. The top channel is pulses that indicate when the hypocretin neuron has fired. And if everything's working right, you should hear pops through the loudspeakers here. The second channel is muscle tone, so it's just reflecting the animal's movement. And the third channel, the one at the bottom, is EEG, so it shows when the animal is aroused and not aroused. So what you'll see is maximal activity is seen when the rat is moving forward, exploring. Also, the neuron's highly active when the rat is grooming or engaging in relaxed eating. I say relaxed eating because you will see an animal approach food and back away from it under those conditions, and even beginning to eat, the cell doesn't fire but very much. But when the animal, if I anthropomorphize, when the animal thinks, oh, this is okay, then the rate goes up. But it's not related to eating per se. My conclusion is it's related to pleasure. And the hypothalamus is known to be involved in emotional regulation going way back. So this is sort of the most interesting part. This rat is asleep. We give the rat the first piece of chicken it's ever seen. That wakes it up, and the rat recoils from what is this, and then he kind of goes back and sniffs some more and sort of tentatively eats. But then after a while, he figures this chicken stuff is okay, and you'll see he eats. And at that point, the cell is firing fairly rapidly. But maybe the main point to be made here is that we're talking about cells that have now been supposedly implicated in the cause of narcolepsy. Well, narcolepsy has two main symptoms. One is sleepiness, and the other is this loss of muscle tone that we call cataplexy. And what you will see, and that's my main point today, is that the activity of these cells does not seem to be particularly correlated with either of these. It's correlated with pleasure in a fairly clear way, but it's hard to see how this causes the symptoms of narcolepsy. So to state this again, it's clear that most hypocretin neurons are lost in human narcolepsy, but it's not clear why the loss of these pleasure-related neurons would cause the cataplexy or sleepiness that define narcolepsy. So I'm going to show a video again. I don't know if advancing it is going to do it because it didn't last time, so I hope the people in AV can help me if this doesn't work. Yeah, there we go. Okay, so there's the rat. The unit signal is at the top. This is frozen. Muscle tone and EEG. So slow wave sleep. And you can see the EEG is high voltage. REM sleep, you can see the EEG is lower voltage. And the popping you're hearing is a hypocretin neuron firing. So you burst right before waking up. And the rat's exploring when it goes, or moving forward, and you'll see grooming. Okay, here's the piece of chicken, and you can anthropomorphize the rat's behavioral state there. See, the unit's completely stopped. But look at the EEG. It's completely activated. So it's not related to EEG. It's related to the emotional correlate. So here he picks at it a little bit. So I describe this as sort of anxious eating. But now he thinks this chicken stuff is all right. Okay, so we had the opportunity to essentially do the same study in humans without putting pieces of chicken in. But, people with epilepsy can usually get their seizures controlled with drugs, but there's a certain population where that does not happen, and these people are at great risk of death from a seizure. And so one way to prevent that is to try and determine where the seizure focus is and surgically remove it. So there's a program that does that at UCLA. What they do is they implant electrodes. This is usually temporal lobe seizures. So they implant electrodes bilaterally, and then the patient just spends a month in the hospital walking around, eating his meals, but being constantly recorded. And when a seizure starts, they look at where did the seizure start, because the devastating thing would be to remove the wrong region. But if you remove only one side, the recovery is pretty complete. I mean, I've spoken to some of these patients. You would never know anything was done to them. And then it does prevent seizures, and they live out normal lives. So as part of this, we were able to put a microdialysis catheter at the tip of these electrodes that they were putting in here to the amygdala and temporal lobe. And cerebrospinal fluid is, artificial cerebrospinal fluid is routed through this semipermeable membrane, and whatever is at high concentration adjacent to the electrode diffuses through there, and then we recover it. We do radioimmunoassay, and we can see how, in this case, the hypocretin level varies from sample to sample. And this is what we found, which is very much like what you saw in the rat. What causes the highest level of hypocretin to be released is, we're breaking this into 15-minute blocks, but when the individual is happy, in Los Angeles, it's when the Dodgers win the game, or when relatives come, you get a very high level. But also quite interesting is the other extreme. I mean, these individuals, not surprisingly, sometimes experience pain from the implant. There were no drugs given in any of these studies during the period we were doing the study. But you can see that the lowest level, even lower than sleep, was when they were in pain. That's the last green thing in the middle of the graph. And then you see not much of a change with eating, not much of a change with sleeping. So, as in the rat, it's not obvious that this is related to waking or alerting or even muscle tone. The other thing I didn't point out is when you saw the rat moving around, the muscle tone is varying a lot. The unit rate is not varying a lot, because sometimes when it's moving, the animal is withdrawing and the unit is completely off. That's what hypocretin cells do. So the bottom line here is these two studies are in good agreement with each other, the human study and the rat study, but it's very hard to understand how they would explain cataplexy or sleepiness, because there isn't any linearity in the relationship to muscle tone or EEG. Okay, so this is the new stuff now, completely new. We have investigated the locus coeruleus. Locus coeruleus is Latin for blue spot. So this is supposedly a blue region of the brainstem. I've looked at many brainstems of many animals, and I've yet to see a blue spot. I think this is a guy 1812 named Wetzel coined the name, and it's a cool name, but I think brown spot would be more accurate. But blue spot is sort of one today, but you'll get to see for yourself. So it's a cluster of neurons in the brainstem containing noradrenaline. These noradrenaline neurons project forward rostrally to the forebrain, and they project caudally, often the same neuron, to the brainstem. Now, the rostral projection, it's well known that there have been many studies showing the locus coeruleus is part of the ascending activating system. That is, part of what keeps you awake is this release of noradrenaline, also called norepinephrine. Locus coeruleus neurons become inactive during REM sleep, causing the loss of muscle tone in REM and preventing dream enactment. And I'm going to connect that to cataplexy because I think our evidence now is that they're related. We wondered if cataplexy might be caused by the inactivity of these neurons during waking. So we recorded from locus coeruleus in our narcoleptic dogs. So this is a midline section of the human brain where it says hypo. That's where the hypocretins are. The hypo in the hypocretin is indicating hypothalamus. And the locus coeruleus, which I have nevertheless put in blue, is in the pontine brainstem, and it has a large anterior-posterior extent. So in these dogs, in this colony that was started a long time ago by Bill Dement, we recorded from 4 locus coeruleus cells simultaneously, which for people who do unit recording will be impressed. It was a lucky day. We had 4 cells, and in A, you see that these cells stop firing, and then they have cataplexy. And conversely, when they start firing, and if they have had cataplexy, they'll start firing before the offset of cataplexy. And in C and D, you can see this averaged over all 4 cells over 20 episodes of cataplexy. And you see there's a very regular diminution of activity before cataplexy onset in locus coeruleus. So I think a good case can be made that locus coeruleus is responsible for cataplexy, or at least the cessation of activity in locus coeruleus. What is already known, which I should have said at the beginning, is we know that locus coeruleus neurons stop firing during the muscle tone suppression of REM sleep. So this isn't REM sleep. This is cataplexy. But it's well established that locus coeruleus stops firing in REM sleep, which is why we wondered whether it had something to do with cataplexy. And of course, locus coeruleus is nowhere near the hypocretin cell field. We were also measuring release of noradrenaline with microdialysis. That's the graph on the right side. So we did that experiment in 1999, if you saw the date on the bottom. And around 2024, we thought maybe we should look at locus coeruleus in these brains. Could have been quicker. So where it says control, that's what locus coeruleus looks like. It's either brown or it's a very, very dark blue. But if you look at narcolepsy, we have 11 brains from people who had narcolepsy. None of them had that brown spot. This is not anything that's stained. This is just, you take a knife and you cut it through the pontine brainstem, and in every control you see this brown spot, and in every narcoleptic it's missing. You can actually, if you look closely in the narcolepsy, you can see there seems to be a little shadow of these cells. I don't know if you can see that, but in almost exactly the same place. And in the microscopic display, control in narcolepsy, you can see the actual cells. They're blue because of the staining now, not because they're in locus coeruleus. It's hematoxylin a stain. So you can see cells in the control, and there are cells in the person with narcolepsy, but not very much. And then we did a test on the number of cells, and it's significantly reduced in narcolepsy, and the size of cells. So apparently the size of the cells is somewhat larger than the average size. That either means that small cells are dying preferentially, or maybe surviving cells are swollen. We'll be exploring that further. But in the middle, you see a plot of cells from control brain and brain of someone with narcolepsy. And you see this tremendous reduction in number in narcolepsy. And it's also most strongly seen in the rostral part of locus coeruleus. This is part near the forebrain. So my conclusion, and my contrarian conclusion now, certainly being part of people cheering on hypocretin neurons as being the total cause of narcolepsy, I now think the one thing that's clearest is the cellular activity in both humans and rats suggests that they're related to pleasure. And there is evidence that people with narcolepsy are significantly more likely to be depressed. And apart from having this chronic disease, one wonders whether the loss of these neurons that we know from both human studies and animal studies seems to be related to pleasure. And so if the pleasure system is damaged, that might explain why this is happening. Now, the locus coeruleus neurons have projections that go forward in the brain to the forebrain, activating the EEG, and also go backward to the brainstem, supporting muscle tone. So I think the loss of ascending axons from locus coeruleus can very well explain sleepiness. There's a huge literature on locus coeruleus role in arousal, and not in relation to narcolepsy, but it's certainly well established that locus coeruleus activity helps keep you alert. So in the absence of locus coeruleus cells, the reduction in locus coeruleus cells, that would be consistent with one of the symptoms of narcolepsy. And the loss of descending axons of locus coeruleus might explain cataplexy in the same way. The locus coeruleus descending projections end up on motor neurons maintaining muscle tone. So if half of them are eliminated, you might think that cataplexy would be more likely. After all, cataplexy is sort of an exaggeration of a normal human response, usually to a positive emotion. There's at least one published paper saying that in every language there's a paper saying something equivalent to weak with laughter, weak with emotion. And if the input that's supporting the muscle tone, if the input to that system is lower, then the same things that would have caused a person to sort of bend over and laugh, then you may have cataplexy. Now, the Honda in 1984 was the first to propose an autoimmune cause for narcolepsy, which I think still stands the test of time. There's a particular HLA haplotype, which is very common to people with narcolepsy. And it may well be that the locus coeruleus and the hypocretin are both damaged in the same episode, or they could be damaged at different times and in different orders. What I can say is that the loss of hypocretin neurons does not cause the loss of locus coeruleus neurons, because we've done that study. That does not happen. So I go back to the drawing, and I know there must be people with narcolepsy in the audience, and I hope you can see this as good news. It's a new damage that's been identified, but it was always there, and it's a new transmitter system that we know a lot about, locus coeruleus, the norepinephrine. There's hundreds, if not thousands, of studies in the neuroscience literature. We know norepinephrine's synthesis pathway. We know the receptors, and it may well be that by looking at noradrenaline and manipulating noradrenaline, you can have a better treatment for narcolepsy than just focusing on hypocretin and giving hypocretin agonists. Okay, so this is the title of the paper. It's in bioRxiv. We have this paper. It's under review now, but if you want to see it, it's on my website. I'll show you the address of the website at the end. The other thing we found, and getting back to this idea that hypocretin neurons are related to pleasure, we kind of stumbled into another very surprising area. Thomas Thaddeus, who works with me and does the histology, came to my office one day and said, you know, and often it's harder to get control brains, because, you know, if you have a neurological disease, you have an incentive to tool your brain for research, but otherwise it's hard to get control brains. So one of my jobs was to find control brains. And he came in and he said, you know, Jerry, there's something very strange about this brain. It has more hypocretin neurons than usual, and they're smaller than usual. And so I thought, well, maybe this person had some sort of strange death that caused this to happen. And, you know, I called the brain bank, and they didn't have the medical records anyway. It's sort of a long story. I happened to be there for another reason, and I noticed that the record storage room was unlocked. So I found this person's records, and there was a note from the guy, and he said, you know, I'm giving my brain because I've been a heroin addict. And it was sort of an electric moment when I found that. And so we gave morphine to mice, and exactly the same changes happened. There were more of the cells, and they were smaller. So I think hypocretin neurons have a major role in opiate addiction, consistent with what I've already said about their pleasure relationship. We've also, now that we have lots of tools for manipulating hypocretin from the geneticists, we've shown that mice that don't have hypocretin neurons do not get addicted. I mean, maybe I should say do not easily get addicted, but in fact what we've seen is they just don't get addicted, period. And we have this paper, it just appeared last year, in which we showed that if you block hypocretin receptors, you can also block opiate addiction without diminishing opiate analgesia. So last year, there were 50,000 opioid overdose deaths in the U.S., and 75% of people who die from opiate overdose, the first opiate administered was for analgesia. And what we've shown in this experiment, there's a lot of experiments, but I know I don't have the time to go into it, and it's not sleep related. but it seems that you can provide the same level of analgesia. We did this in mice, and no addiction. So I think maybe in the future we'll mix morphine with suvorexant and save some lives. So analgesia is not diminished at all. So all the papers I've talked about are on that website. If you want, you can take a picture, and I'm happy to answer your questions at my email address.